We completed glycolysis and Krebs cycle. Electron transport chain we have to explore. Correct, no? Electron transport chain is for what purpose? What do we need to achieve through electron transport chain? What is the cell trying to achieve through electron transport chain? Purpose of electron transport chain is to transform the energy that is stored in the high energy compounds NADH and FADH2 into the energy of ATP. ATP production is the purpose of electron transport chain. That is the purpose. Design, we have seen complexes. Different complexes are there which will allow transportation of electrons or movement of electrons from one to the other. Why should there be these complexes? Why can't it be just NADPH is broken down, energy is released, bring ATP and ADP and phosphate together and produce ATP just like other enzymes do? Why not that way? Why did it not happen that way? Think of some logic. Everything that is existing in nature exists for an evolutionary reason. We have seen substrate level phosphorylation happening. Yes or no? Pyruvate kinase. Phosphoenol pyruvate is broken down by pyruvate kinase. Pyruvate is produced. Breaking down of phosphoenol pyruvate released one phosphate group also, and that bond breakage released energy also. That energy was used by the enzyme pyruvate kinase in fixing ATP and fixing ADP and phosphate together. ATP was formed. Why couldn't that same process happen? Why do we need to have a series of electron carriers and at the end of it there is an ATP synthase? You create a proton gradient, create an electron gradient. Why should all this be done? And this is making it necessary for oxygen also to be present. If oxygen is not there, then this process will not happen. Majority of the energy dependency for living organisms has come from ATP, is on ATP. Living organisms, aerobic organisms are depending on ATP. If this was not there and all phosphorylation would have happened at the substrate level, then there would be no problem only, no? We would have not required oxygen also. The same process could have been done elsewhere with different enzymes. Why are we having this mechanism? Think of an evolutionary significance of that. It is not like it has been created. It has evolved to be what it is today. Why did it evolve to exist in the way that it is today? Anything that has evolved in function would have evolved as a response to a challenge, would have evolved as an advancement or an improvisation of the existing process. So if we have mitochondria mediating the process of converting energy present in NADPH and FADH2 into ATP through this electron transport, creating proton gradient and all that, it means that it has given advantage. That is why it has survived. We see in Darwin's theory, no? Darwin's theory is survival of the fittest. So those who have an adaptation or that process which has an adaptation which leads to advantage will eventually exist, and that which is causing not so much of an advantage or causing a disadvantage will eventually go away. So survival of the fittest is not like the fitter ones are beating up and killing the weaker ones. Eventually, the probability of replication of a successful process is higher than the probability of replication of a not so successful process. We cannot say failed process. If something is existing, it has been useful for performing a particular function to an extent. You get something which is better. In comparison to that, this may appear a failure, right? Lactic acid metabolism, lactate metabolism. If the pyruvate goes into lactate pathway, it gives you overall one glucose will give you 4 ATPs. One glucose will give you 2 ATPs. But when you look at it in an aerobic pathway, one glucose will give you 38 ATPs. So compared to aerobic respiration, lactate metabolism based anaerobic respiration appears to be a failure. But in the absence of aerobic respiration, this is still doing its job of providing ATPs, no? This is still doing its job of allowing the pathway for breakdown of glucose in the production of ATP. So success and failure are relative. If what we have right now, the electron transport chain based ATP generation, is the most successful model of energy production in living systems at this point of time. We may be going through, as living organisms, we may be going through further evolution, which may be another 10 million years down the line have something else. We will not know that. Evolution is looked at in a time frame of millions of years. In one generation, we will not be able to notice. We might be going through, but what is being useful for us is the most productive process as of today. Electron transport chain. What is the advantage? Think logically. Why did evolution choose this process? Because it is hassle. You have to create so many electron carriers in multiple complexes. Proton gradient has to be created. It is increasing work. But is the increased work worth it in fulfilling the purpose? It should be. That is why the process exists. So purpose needs to be fulfilled. Doesn't matter whether the work is more or less. Similarly with kidneys. I have told you this earlier also. Kidneys will filter out everything that can pass out to the mechanical filter and take back everything that is required. 99% of everything that is filtered is taken back. 1% is excreted. Laborious process, but the purpose is served. That is why it exists. Nature wants the purpose to be served. So effectiveness is nature's way, not efficiency. If there is efficiency, great. But the work should happen. Effectiveness is important, not efficiency. So in the situation, this is the most efficient process. Anything else would not be effective at all. If you try to improve the efficiency and try to make it something else, the other processes have not been able to survive. My question is, why should NADPH donate electrons flow through these complexes and then finally ATP is produced? Why should it not be just break down NADPH, energy is released, use that energy to make ATP? Like we see it in other reactions, no? Why produce NADPH in the first place? Why can't you produce directly ATP in the metabolic reactions? Right? 3-phosphoglycerate to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate to 2-phosphoglycerate. In that process, we have NADPH being produced. NADPH is being produced. Why ATP only could have been produced, no directly? Why produce NADPH, FADH2 and take it through electron transport chain to make it ATP again? Why not produce ATP directly? Because when you go from one process to another process, there is more work is increasing, and also there is a possibility of further loss of energy. So two questions: why produce NADPH, FADH2 in the first place? And second one, why take it through a series of electron transport reactions to convert that energy into ATP, when you could have easily done it by breaking down that NADPH and using the energy of breaking down the bonds to make the bonds between ADP and phosphate produce ATP? No? In a kinase reaction, yeah.
If we do that, then most of the energy is released as heat. So if we stepwise, so that we can use maximum of the energy.
Okay. The direction of thinking is okay. You are on the same road. Okay. Anybody else wants to think? Okay, I'll ask you more questions. When a bond is broken, energy is released. And that energy is fixed in some other bond. Okay. Let us say the bond between ADP and phosphate, between two phosphate, the phosphate bonds in ATP has, wake up. See, some basic ground rules, okay. I don't care how you manage your break time. Manage your break time to sleep well, rest well, so that in the study and learning time you are awake. You cannot say I am tired so I will be sleeping in the class or during study time. No. You can't do anything that you are not allowed to do in an examination hall in this room. Either during study time or during class time. Alright? Not because I want you to go through some torture. It is a very important part of training your body and mind. Not easy to sit with attention straight on a chair at a table for 11.5 hours in a day. But that training you have to do. How you will be able to manage that is by managing your rest properly. As soon as you finish the last session, go and sleep. During the breaks through the day, sleep. Learn to sleep. Alright? Otherwise, losing attention here, sleeping in this room, you will be noted, you will be made to stand, or you will be made to go out, wash your face, all that. Every time somebody is sleeping here, they will be made to be known to everybody that somebody was sleeping. Alright. Practice, practice, practice. Training the body to listen to your mind is the most important thing that needs to happen. You have done significantly well in 1 month. We have another month for adaptation. Practice to sit here. In this month, focus more on resting. Rest well so that you can stay awake when you are supposed to be awake. Alright? If you feel sleepy, go wash your face, walk twice in the corridor, have a glass of water, come back and sit. Go out when you go out to wash your face, stretch your body and come back and sleep. In the morning after your first session, in that 1 hour, 10-15 minutes of sleep is necessary. Because you would have woken up at 5 o'clock, done your cycle of yoga, 1 session of learning, body will require rest. So you are not sleeping during that time. That is the problem. No sleeping, no taking breaks during scheduled sessions. No washroom breaks. Madam, please note. No washroom breaks. 3 hours is not a time in which you will require breaks in between. Finish it before you come here. If you finish your washroom breaks before you come to the session, and then even then if you need a break in between the session, that means there is some disease. There might be a urinary tract infection or gastroenteritis. Alright. If not, if that is the case, we will treat. We will treat it. We will get it evaluated and treat it. That's okay. But otherwise, under normal conditions, you should not be getting up within 3 hours. So 2 sessions of 3 hours is to make you get comfortable with sitting at one place for 3 hours. You know that, no? It's 3 hours, 35 minutes that you have to be in one place during exams. Your body should be the last thing that troubles you at that time. And all that attention span is 7 minutes, 10 minutes, 20 minutes. I don't subscribe to all that. How much ever you train, that much more will be your attention. Train your body and mind, the body and mind will listen to you. Alright? So, consider this space as being similar to an examination hall. You are not supposed to do anything in this space which you are not allowed to do in an examination hall. Alright? Practice, practice. It is not easy. It is not easy, but train yourself well. Managing rest properly will help you to perform. You don't rest well, then you can't work well. Alright? Learn to do things at those respective times. The breaks during the day post breakfast, post lunch and post dinner is for resting. 1.5 hours you have, 4 to 5:30. That is the time when you play, you socialize, you meet, you talk to your friends, gossip, take out all of your anger on me, on anybody, with each other. Alright. There used to be a postgraduate in the Department of Medicine when we were in our final year MBBS. Lady. She used to be very rude to us. And we can't do anything because postgraduates have a control on how our final clinical part of the exams go. So some of our classmates decided that we have to somehow take out our anger on her. She used to come in a scooty. Now you know what can you do? Alright. Unfortunately, her scooty used to have a lot of punctures and a lot of seat damage. I don't know what used to happen, but that is what she used to experience multiple times. Then gradually she started becoming more kind towards medical students and interns. Any correlation? I don't know. Alright. So all those things you can't do, but 4 to 5:30 is the time when you can do all your fun, socializing, play, all of that is there. But post breakfast, post lunch, post dinner, that time should be utilized for rest. Alright, okay. So, coming back to this. Energy is released when something is broken, energy is fixed in the bond between two substances. The bond between two phosphates can hold certain amount of energy. That's a high energy bond. NADPH and FADH2 can harness more energy than what ATP can. They are bigger currencies. If ATP is like a 10 rupee note, NADPH is like a 50 rupee note. FADH2 is like a 30 rupees note. Alright. If you are using the energy released by a process which is releasing so much energy and using that to store in the form of ATP during metabolic reactions, ATP will be produced, but a lot of energy will be wasted, lost. To increase energy harnessing efficiency, to increase the efficiency of harnessing energy, when a chemical reaction is happening, when a metabolic reaction is happening and the breakdown of something is happening and it is releasing more energy, it makes sense to store that energy in a bigger, higher energy compound. In a compound which can hold more energy than ATP. So NADPH can hold more energy than ATP. Alright. That is why when NADPH goes through the electron transport chain, one NADPH will give you 3 ATPs. That means NADPH is harnessing energy equivalent to more than 3 ATPs. Isn't it? Because in the process of transferring energy from one state to another state, there will be loss of energy also. So instead of NADPH, if you had produced ATP only during that metabolic reaction, then the energy would have been wasted because ATP would be produced, but what will happen to the excess energy that was released in the breakdown of a particular compound, in a particular catabolic reaction? Isn't it? Understanding? So NADPH and FADH2 is required. Okay, but why electron transport chain? Why electron transport chain? Electrons also can gain energy. When you supply energy to electrons, what will happen to the electrons? The electrons will get excited, they go into a higher energy state. When electrons pass on from a higher energy state to the next one in the line, electrons are coming down the energy state. When they go from a higher energy state to the next lower energy state, energy is being released. Energy is being released. Now, what is this energy used for in the electron transport mechanism? The energy released when electrons flow from higher energy state to lower energy state is used for doing what? Pumping out protons. It is used to pump out protons. Now, why protons? Electrons and protons go hand in hand in biological systems. When you are exciting electrons, when you are breaking down electrons, protons are also broken off, released. So you are having protons. Why not something else? Electron and proton go hand in hand. Okay. So protons are pumped out. Then you have an enzyme, ATP synthase complex, an enzyme complex, which has a mechanism of producing ATP when protons flow through it. So your task of while you are moving electrons from one point to another point, when electrons are being flown through the system, it is designed in such a way that by the time all the energy that was given by NADPH breakdown to excite the electrons has been utilized for pushing out maximum proton ions into the intermembrane space. Now ATP synthase being the only one which can allow it to come back, it will flow through the ATP synthase complex back into the matrix, ensuring ATP is produced every time protons flow through it. So one chemical reaction, one metabolic reaction where something was broken and energy was released, more energy was released, so it was harnessed in NADPH or FADH2 if the energy was little lesser than NADPH but more than ATP. Then this mechanism allows you to transform that energy into ATP. If not for this process, the same metabolic reaction would have given you 1 ATP. Energy was released, ATP could have been produced. It would have been 1 ATP. But because you increased this, because you installed a process of harnessing that high energy into multiple ATPs, though the work went on to become more, the utility of breaking down a particular organic compound in the cell for the purpose of harnessing energy is served better. Does that mean we have efficient mechanism of harnessing energy? We don't have an efficient mechanism of harnessing energy, but we have the most effective mechanism of harnessing energy. So as we move forward, just like our internal combustion engines are becoming better and better in their fuel utilization efficiency, our systems also might continue to evolve and become better at utilizing the energy. But right now, this is the mechanism which will allow us to harness the maximum possible energy. Possibilities can change, possibilities increase based on the evolution. Evolution creates possibilities. Necessity leads to evolution, evolution creates possibilities, possibilities leads to enhancement of function, enhancement of function leads to gaining advantage, cycle keeps on going upwards. It's a virtuous cycle. Alright? So, yes.
Is NADPH and NADH are same?
Same. Yes, same in terms of harnessing energy. Chemically they are different. Nicotinamide, NAD is nicotinamide adenine diphosphate. NADP is nicotinamide adenine diphosphate. NAD, NADP are representation of the same thing. Okay. H plus. It's generally two hydrogen ions that it holds on to. NADP H plus H plus. So one is in an ionic form, ionized form, and one is bound to the NAD complex. Alright. Okay. Is this clear? The basis of electron transport chain. Why should we have it? Purpose of it. See, whether it is science or history, mathematics, everything has a story. It's a philosophy. You have to understand the story behind it, you have to understand the philosophy behind it, then the specifics becomes very easy. Now we will just go through, help you to fix it in multiple perspectives. Electron transport chain, or it is also called oxidative phosphorylation. Phosphorylation is attaching a phosphate group to something. Generally, phosphorylation in terms of energy we talk about with reference to making ATP. So phosphorylation reactions can be like this one, 3-phosphoglycerate becoming 1,3-bisphosphoglycerate is also phosphorylation only. You are adding phosphate group to it. Alright. But when it comes to energy dynamics, for the sake of simplicity, we will refer to it as ATP producing reactions. So there are three types of phosphorylation reactions that you can see in living systems. Number one is photophosphorylation, seen in autotrophs, seen in photoautotrophs, those organisms which will utilize sunlight, energy of sunlight to harness it into ATP, photophosphorylation. Second is phosphorylation happening during metabolic reactions, substrate level phosphorylation, when a substrate is being broken down and the energy is used for producing ATP, that is substrate level phosphorylation. Third one is oxidative phosphorylation happening in mitochondria, when oxygen is utilized in the process of making ATP. Oxygen dependent ATP production is oxidative phosphorylation. Alright. Photophosphorylation and oxidative phosphorylation are dependent on electron transport chains. Electron transport chain is the basis of photophosphorylation and oxidative phosphorylation. Substrate level phosphorylation are enzyme dependent. Kinases. During chemical reactions, the energy is harnessed into ATP. So oxidative phosphorylation, photophosphorylation depends on electron transport. Substrate level phosphorylation depends on the enzyme's ability to phosphorylate. Not dependent on energy or not dependent on oxygen or sunlight. Photophosphorylation does not require oxygen for phosphorylation reactions. Oxidative phosphorylation requires oxygen. Clear? Now, transportation of electrons is associated with a very important coupled reactions. Reduction, oxidation. Oxidation, reduction reactions. Now, when you have A, you are removing electron from that. What will happen to A in terms of its charge? It becomes negative or positive? If you remove electrons from something, does that become negatively charged or positively charged? Positively charged. You remove electron, there is positive charge. Electrons that are removed, or there is B, you are giving electrons to B. What will happen to B? Negatively charged. Alright? Keep it this one. Now I am talking about positive and negative charge in terms of removal of electron or addition of electron. If you remove electron, something becomes positively charged. If you add electron, something becomes negatively charged. There is no confusion with that. Now, if you remove proton from something, that something will become positively charged or negatively charged? What will happen to something that from which protons are removed? It becomes negatively charged. So you are adding electrons to A, you are removing protons from B. Both are negatively charged now. If you are adding protons to something, what will its charge become? Positive or negative? Positive. Now I will tell you, removal of electrons is oxidation, addition of electrons into something is reduction. If you add electrons, it is reduction. If you remove electrons, it is oxidation. So can I say something which is negatively charged is reduced? Is something that is negatively charged reduced? Is it in a reduced state? If you add electrons, it is reduction. Adding electrons will make something negatively charged. Can I state it as anything that is negatively charged is reduced? Adding electrons to something, addition of electrons is reduction. Counter statement I am giving is anything that is negatively charged is reduced. True or false? No? Yes? You are saying no? Those of you who agree, anything which is negatively charged is in a reduced state, raise your hands. If you feel anything that is negatively charged is in a reduced state, raise your hands. What if you feel so? Yes, raise your hands. Those who say no, raise your hands. No. Okay, can you explain? Wait, we'll complete the options. Anyways, only one person has voted for no. Those who say no idea what you are speaking about, I don't know. Raise your hands. Those of you who say, I am not sure, confused, it may be reduced, may not be reduced, raise your hands. Okay. I will safely assume at this stage that nobody will choose the fifth option. I won't say. Anybody wants to choose that? You are choosing fifth option? No. Okay. Alright. This is a basic confusion that people will have with respect to oxidation and reduction. Remember, oxidation and reduction is related to electrons. If negative charge is because of addition of electrons, then it is reduction. If positive charge is because of removal of electrons, then it is oxidation. Alright? Addition of protons will make something positively charged, but it is not oxidation. Removal of protons will make something negatively charged, but it is not reduction. Anything that is negatively charged may or may not be in a reduced state. It will be in a reduced state if the reduction, if the negative charge is because of addition of electrons. Anything that is positively charged is not necessarily in a state of oxidation. It would be in a state of oxidation only if the positive charge is because of removal of electrons. So with respect to oxidation and reduction, remember it is dependent on electron, nothing else. Alright, now we'll see this. In the context of redox reactions, the term oxidized and reduced specifically refer to the gain or loss of electrons and not protons. Oxidation typically involves the loss of electrons, leading to an increase in positive charge or a decrease in negative charge. Reduction involves the gain of electrons, leading to an increase in negative charge or a decrease in positive charge. Alright, if negative charge increases because of electron being added, then it is reduction. The confusion arises because the loss of protons can indeed make a molecule more negatively charged. However, this doesn't necessarily mean the molecule is reduced. See, a conversational book is a teacher in itself. You don't need an interpreter. However, this doesn't necessarily mean the molecule is reduced. The redox state, oxidized or reduced, is determined by the electron transfer, not by proton transfer. Be very clear about this. It can help you in chemistry also. Corrected understanding, losing H plus ion decreases the positive charge of a substance, making it more negatively charged. However, this alone doesn't mean the substance is reduced unless it also gains electrons. Alright. Just by losing protons, you gain negative charge. That is not necessarily reduction unless you also have gained electrons. Okay. If you are losing protons and gaining electrons simultaneously, is it reduction? You are losing protons and gaining electron simultaneously in the same breath. Is it reduction? It is reduction. You are losing electrons and gaining protons. Is it oxidation? It is oxidation. But if you are only losing protons or gaining protons, that is not considered to be oxidation or reduction. Alright? Gaining protons. This increases the positive charge of a substance, making it more positively charged. Again, this doesn't mean the substance is oxidized unless it also loses electrons. Okay? Now sometimes it can be confusion between what is reduction, what is oxidation, losing electrons or gaining electrons. That is also possible, no? Like the confusion between what is the correct spelling of friend. What is the correct spelling of friend? What is the correct spelling of friend? F-R-I-E-N-D. F-R-E-I-N-D? Is it wrong? Is it wrong? I used to have this confusion for a long time, which continues to exist as a confusion even now. So I no longer bother about what I write, FREIND or FRIEND, and I have checked it, both are correct spellings. Alright. So gaining electron, losing electron, we understood, okay. What should we consider as reduction? What should we consider as oxidation? Okay. Equate electrons to negative attitude, negative mindset. Equate electrons to negative mindset. Oxygen is energy, positivity. Correct, no? If you have more negative mindset, what will happen to your personality? It will reduce. If you have more negative mindset, your personality will reduce. So you have more negative charge, it is reduction. If you lose negativity, you are more positive. If you lose negativity, you are more positive. Oxygen. It gives you oxygen for your life. Oxidation. Alright. Some nonsensical jugads. You are confused about remembering something, it's a memory technique also. You have to relate technical things to the most observed things. Then it becomes easy to remember. It becomes easy to remember. Alright. There are two more such things that I use for remembering list sequence. Like let's say cranial nerves. You have to remember cranial nerves in a sequence. You have 12 cranial nerves. You have to remember all the nerves in a sequence. You can't remember it in a jumbled up order. Excuse me. Similarly, you have essential amino acids, 10 essential amino acids. You have to remember that in a sequence. Otherwise, you will not be able to recall. Questions will be based on directly recalling the amino acid sequence. Which of the following is an essential amino acid? Can anybody tell me what are the 10 essential amino acids? How many amino acids are required for human body to function normally? 20 amino acids. 10 are produced by our body. 10 has to be supplied in the diet. What can be produced is not necessarily to be supplied, so it is not essential. What has to be supplied is essential. Alright, how to remember? Aladdin will help. You know the story of Aladdin? Aladdin got some gift by which he could go and meet the princess. What was the gift which he used to travel to meet the princess? Magic? Carpet. Carpet. What will the magic carpet do? It will fly. Give me some other term for carpet. Huh? Mat. That's all. 10 essential amino acids is mat will fly. But during Aladdin's time, English was not so well developed. There will be spelling mistakes. MATT, VILL, PHLY. Alright. Methionine, arginine, threonine, tryptophan. VILL. Valine, isoleucine, leucine. PHLY. Phenylalanine, lysine. Phenylalanine, lysine. Histidine. Phenylalanine, histidine, lysine. 10 over no? Methionine, arginine, threonine, tryptophan, MAT. Valine, V. IL, isoleucine. L, leucine. 4 plus 3, 7. Phenylalanine, P. H is histidine. 9. LY is lysine. MAT will fly. 10 amino acids you remember. Alright. So, such correlations you should find. Alright. So, I have tabulated this redox reaction tabulation. Action, is it oxidized or reduced? And some explanation. If something loses electrons, oxidized, yes. Reduced, no. Losing electrons increases positive charge, oxidation. Same, gaining electrons, reduction. Gaining electrons increases negative charge. Loses protons, it is neither oxidized nor reduced. Losing protons makes the substance more negatively charged, but it doesn't equate to reduction directly. Similarly with gaining protons, becomes positively charged. It might be oxidation if it is due to electron loss. Definitely not reduction if it is positively charged. If something is negatively charged, it might be reduction if it is due to electron gain. It certainly is not oxidation. Alright? This table will help you understand oxidation reduction in three perspectives. In the perspective of gaining or losing electrons, gaining or losing protons, and in the net charge a particular substance has. Alright? All three are interrelated. Next we'll go. Loses electrons, gains electrons, loses protons, gains protons, become positive charge, becomes negative charge. Purpose of electron transport chain. We already discussed that ATP production, formation of water. Which complex is associated with formation of water? Complex 4. Complex 4 is associated with formation of water. What do we call that water? Metabolic water. We call that metabolic water. What all are essential there at complex 4 for the formation of water? Protons, H plus, hydrogen ions, oxygen and electrons. Electrons are coming from the electron transport chain. Oxygen is coming from blood. In the cell it is available. And the hydrogen ions are already formed within the mitochondria there. Then regeneration of NAD plus and FAD. NADH and FADH2 have been produced during metabolic reactions. NAD plus and FAD should be made available for the same kind of reactions to again take energy. No, you cannot keep NADH and NAD and FAD in the same bound state. You have to release it for participating in further reactions. Where does the ETC operate? On the inner mitochondrial membrane, designed for ETC to operate. Protein complexes. The electron transport chain consists of a series of protein complexes 1, 2, 3, 4 embedded in the inner mitochondrial membrane. We have already discussed all this. I'll just read through. These complexes are arranged in a specific sequence to facilitate the stepwise transfer of electrons. Complex 1. What is happening there? NADH is being broken down to NAD plus H plus. Correct? Because NADH is being dehydrogenazed, hydrogen is being removed, the complex is referred to as NADH dehydrogenase complex. NADH dehydrogenase complex. It is not just a single enzyme, there are other substances also inside that. We will see that. Accepts electron from NADH and pumps protons across the membrane. Complex 1. Complex 2 is called succinate dehydrogenase. Accepts electrons from FADH2 but does not pump protons. We have seen this already. Complex 2 is not present throughout the membrane, through the membrane. Alright, it is partially present on only the inner side of the mitochondrial membrane. Complex 3, it's called cytochrome BC1 complex. Transfers electrons from ubiquinone to cytochrome C and pumps protons. Forget about these things right now. Cytochrome B, cytochrome C1, ubiquinone, all of that let it be. It transfers electrons further and it also pumps protons. Complex 4, cytochrome C oxidase. Transfers electrons to oxygen, reducing it to water and pumps protons. Alright. Electron carriers. So what do we have in the electron transport chain? The design protein complexes are there, 1, 2, 3, 4. Then we have electron carriers. Small molecules like ubiquinone and cytochrome C shuttle electrons between the protein complexes. Ubiquinone and cytochrome C are present between complexes. Between 1, 2 and 3 is ubiquinone. Between 3 and 4 is cytochrome C. So proton gradient. The electron transport chain is designed to pump protons from the mitochondrial matrix into the intermembrane space, creating a proton gradient. It's also called proton motive force. Motive force means force for the movement of protons. This gradient is crucial for ATP synthesis. We have already discussed this. At any stage, if anybody is finding it difficult to understand, stop me. Since we have already discussed all this, I am just running through it. Alright? Is it clear so far? Yeah. Complex 5 is considered separately as ATP synthase. Alright. The other four complexes are associated with electron transport. ATP synthase is associated with ATP production by letting the flow of protons. So, some references will talk about ATP synthase as a fifth complex. But keeping ATP synthase as a separate entity and not refer to it as an electron transport complex is more better for clear understanding. Alright. 1, 2, 3, 4 are associated with electron movement. ATP synthase is not. Though it is also a complex. If somebody is referring to it as complex 5, read it as ATP synthase. Nothing wrong in referring it to as 5 because this entire process has 5 complex of proteins. 1, 2, 3, 4 and ATP synthase separately is better. ATP synthase, a specialized enzyme called ATP synthase uses the energy stored in the proton gradient to convert ADP and inorganic phosphate into ATP as protons flow back into the mitochondrial matrix. Compartmentalization. The design of the mitochondria with its double membrane structure is crucial for the electron transport chain's function. Inner mitochondrial membranes impermeability to protons ensures that the proton gradient is maintained, which is essential for ATP production. Okay. We don't want protons to just keep moving from the inner membrane to the intermembrane space, from the matrix to the intermembrane space. It has to move only when transported. So the inner mitochondrial membrane is impermeable to protons. So design is so intricate. So purpose, location, design, all of that we have summarized. This design ensures that the cell efficiently produces ATP while maintaining the flow of electrons and protons in a controlled manner. Coordinated functioning of complex 4 and ATP synthase. Complex 4 and ATP synthase operate in coordination, but they have distinct roles within the electron transport chain. Their coordinated function is crucial for overall process of ATP production and cell respiration. Why should there be coordination between ATP synthase and complex 4? Complex 4 is the one which is providing possibility for oxygen to accept the electrons. That is crucial for ensuring the proton gradient. So, complex 4 malfunctioning will lead to malfunctioning of ATP synthase complex. Alright. How they coordinate? Role of complex 4. Electron transfer and proton pumping, role of ATP synthase, utilizing the proton gradient and ATP production. Proton gradient dependency, I told you that, and sequential operation of electron transport. Okay. So just a diagrammatic representation of this thing. Now this will make sense. After all this discussion, the diagram will give you a visualization of that. Alright. What is this space? This one, this matchbox matchsticks. What is this? Huh? Inner mitochondrial membrane. This is outer mitochondrial membrane. So this is mitochondrial matrix. Outside of this is cytoplasm. So this is our space. What is this space? Intermembrane space. You see complex 1, complex 2, 3, 4 ATP synthase. Direction of proton movement, electron transport, NADH becomes NAD, electrons are released, passes on. What is this? Can you tell me what is this? 1 and 2 are transferring electrons to this, 3 is transferring electrons to this. So what is this and this? No, these things. This one. Oh. Oi. No. Okay. This one. This And this. We already discussed this. I told you this. Ubiquinone and cytochrome C. You see this? Ubiquinone, these are fixed complexes. 1, 2, 3, 4 are fixed complexes. Ubiquinone and cytochrome C are shuttlers. They shuttle. Alright. This fellow goes to this to carry electrons and delivers to 3. If this one is giving, it will carry from here. This will go here, take it here and move. It keeps moving. They are shuttlers. They are shuttlers. Who are shuttlers in the entire electron transport chain? Ubiquinone and cytochrome C. Rest all are fixed in their respective places. They don't move. 1, 2, 3, 4 are fixed. This is ATP synthase. You see this? As this flows, it has a kind of rotatory movement inside the enzyme complex, which will bring ADP and phosphate together to make ATP. Proton is flowing backwards. Is this clear? See here, Krebs cycle is also mentioned. NADP and FADH2 are produced. No. Alright, do you understand this diagram? You can watch it, see it, observe, understand. It should be the other way. Anyways. Electron donor is NADH at complex 1. Electron acceptor from complex 1 will be ubiquinone. Same. Electron acceptor from complex 2 will be ubiquinone. Complex 3, from complex 3 electron acceptor will be cytochrome C. From complex 4 electron acceptor will be oxygen. There are chemicals, electron carriers in between also. We will see that. But right now we will look at complexes. Name the complexes. Complex 1 is referred to as NADH, dehydrogenase. Complex 2 is referred to as what? Succinate, dehydrogenase. Complex 3 is referred to as cytochrome BC1 complex. Complex 4 is referred to as cytochrome oxidase. Why cytochrome oxidase? The dehydrogenase is understood. It is dehydrogenating particular electron donors. So named after that. Cytochrome BC1 complex is also understood because there are cytochrome B and cytochrome C inside that complex, which will help in electron transport. So it is referring to the components of the complex. Why cytochrome oxidase for complex 4? When complex 1 is donating electrons to ubiquinone, ubiquinone is getting reduced, complex 1 is getting oxidized. Next one, ubiquinone to complex 3, ubiquinone is getting oxidized, complex 3 is getting reduced. Complex 3 to cytochrome C, cytochrome C is getting reduced, complex 3 is getting oxidized. Cytochrome C to complex 4. When complex 4 receives electron from cytochrome C, what will happen to cytochrome C? Oxidized. So can I say complex 4 is responsible for oxidation of cytochrome because it takes away electrons from complex cytochrome? Why is the complex 4 referred to as cytochrome oxidase? Because it is receiving electrons from cytochrome, thereby leading to the oxidation of cytochrome. In the same way, can we say complex 1 as NAD oxidase? We can say that, no? Right? NADP, NADH oxidase. Complex 2 as FADH oxidase. Ubiquinone as complex 1 oxidase. Complex 3 as ubiquinone oxidase. Cytochrome as cytochrome C as complex 3 oxidase. You can call whatever you want to call, but there are names given to it. We will remember those names. But it's a logic behind the naming of these complexes. Complex 4 is named as cytochrome oxidase because it is oxidizing cytochrome when it receives electrons. That's all. So tell me the sequence of electron flow. If NADH is the electron donor, tell me the sequence of electron flow from NADH to oxygen. Start. NADH. Everybody should Tell it. NADH, complex 1, ubiquinone, complex 3, cytochrome C, complex 4, then oxygen. If FADH2 is the electron donor, tell me the sequence of electron flow. FADH2, complex 2, ubiquinone, complex 3, cytochrome C, complex 4, oxygen. That's all. Ubiquinone is the junction. Question can be in electron transport chain of electron transports in the mitochondria of eukaryotic cells. The junction of meeting of the electrons coming from different electron donors is, which is the junction where the electrons of different donors are actually coming together? Ubiquinone. Ubiquinone is the one which is the junction, that is the junctional compound. Alright, questions can be anything. Questions can be anything related to these concepts. Alright, we'll go further. I have given further different ways of writing down the same thing. What is an electron donor, electron acceptor, what is a complex, proton pumping, ATP produced. Okay. Complex 1, 3 and 4 pump protons, complex 2 doesn't. Movement of protons happens across what membrane in mitochondria? Inner mitochondrial membrane. Where do the protons move from and into where? Move from mitochondrial matrix into intermembrane space. Is the transfer of electrons providing energy for the movement of protons? Is the transfer of electrons providing energy for the movement of protons? Yes. How are NADH and FADH2 ionized by the complexes? NADH dehydrogenase is an enzyme which will break down NADH and ionize it. Similarly, succinate dehydrogenase does it at complex 2. Details of ATP synthase. How is ATP produced and how is water produced? ATP production. How is ATP produced? Protons from the intermembrane space flow into the mitochondrial matrix through the ATP synthase. That flow of protons will provide energy for the combination of ADP and phosphate to produce ATP. Alright. Water production. At complex 4, which is cytochrome oxidase, the terminal electron acceptor is oxygen. Proton, oxygen and electrons combine together to form water. That is the metabolic water. Why should we consider NADH as equivalent to 3 ATP while FADH2 is as 2 ATP? That is already answered. Why? Why NADH is equal to 3 ATP and FADH2 is equal to 2 ATP? Electron donated, that answer you should not use. Talk in terms of ETC. Electrons donated by NADH2 leads to the expulsion of 3 protons in complex 1, 3 and 4, which when coming back through ATP synthase leads to the production of 3 ATPs. Whereas electrons donated by FADH2 leads to the expulsion of 2 protons, which while coming back leads to the production of 2 ATPs. Proton expulsion by the electrons donated by FADH2 happens in which complexes? Proton expulsion by the energy of electrons donated by complex 2, FADH2 happens in which complexes? 3 and 4. Proton expulsion by the flow of electrons donated by NADH happens in which complexes? 1, 3, 4. Where do we need oxygen in this process? Also, why do we die if there is no oxygen? Already answered. Where do we need oxygen? Complex 4. Why do we die if we don't have oxygen? No terminal electron acceptor, no ATP synthesis. Okay, done. Now you have become masters of respiration, glycolysis, Krebs cycle and electron transport chain. Lot of questions you will be able to answer. Revisit all the questions which have appeared from this topic in the papers that you have attempted so far and see the logic that will emerge in your mind to arrive at answers for these questions. Alright, good. Any questions? Anything? Any clarifications? Do you want this notes? The soft copy? Now only? Okay. Then you will stop making your post-test analysis notes. Huh? If I start giving you my notes, then you will stop making your notes. No? Alright, we'll see. Okay. During seed germination, its stored food is mobilized by. What will mobilize the stored food during seed mobilization? All the four given, there is answer visible. You should already be knowing because you already have seen this. Okay. How many plant hormones do we study? Which of the 5 is missing in this? Auxin is missing. So 5 plant hormones we study. 5 plant hormones we study in 2 or 3 categories. Growth promoters, growth inhibitors, and fruit ripening hormone, ethylene, gaseous hormone. Alright. What is the purpose of hormones? We also have hormones. What do hormones do? How are hormones different from enzymes? There are 2 sets of chemicals that we encounter, even whether it is plants or human beings. We encounter 2 sets of chemicals which influence the body's functioning. Enzymes and hormones. What are enzymes and what are hormones? What do enzymes do? What hormones do?
According to me, enzymes are produced with a duct, and hormones are low protein and plasma.
Plants?
Enzymes are produced in large amount and hormones are produced in trace amount.
Okay. What about enzymes produced in very small amount? We can call them hormones. Okay.
Enzymes are involved in reactions for a short duration.
Enzymes are?
Involved in reactions for a small duration. For short
Okay.
duration. Hormones are involved in long
Continuous action. Okay. Okay. Now I'm not agreeing or disagreeing with anything you say. I just want you to say whatever you feel. Yes. So she says enzymes are involved in short duration action. Hormones are involved in long duration action. Okay. Then
hormones are important for growth and development, while the enzymes are just for
hormones are important for growth and development, while enzymes are
for metabolic reactions.
for metabolic reactions. Okay. Growth and development also is a consequence of metabolic reactions. Correct, no? All things that happen in our body, whether it is growth, development, cellular metabolism, everything is metabolic reaction only. Hormones are also produced because of metabolic reactions within the hormone producing cells. Though you are right, enzymes are involved in metabolic reactions, they mediate metabolic reactions. Hormones are involved in growth and development. Can you elaborate a little bit on that? My conflict with this is even growth and development are a consequence of metabolic reactions. So what is hormone doing? What is hormone doing? Who built this building? Don't worry about the name. Who built this building? Huh? Who built it? Mason. Okay. Does the mason build? Okay, masons built this building. At different levels there are executive people. People who execute instruction, execute action. Let's consider functions in our body is like this building. Functions in our body is like this building. You have to build the building. For what do you need first for making the building? Blueprint. Building will happen according to the blueprint only. Whatever material you use, you are trying to give shape to the blueprint. So that blueprint is our genetic material, DNA. Blueprint is our DNA, genetic material. Alright. Based on the blueprint is like the architect plan. Now that architect plan has to be translated into working principles. You need to make a structural diagram, you need to make an electrical diagram, you need to make a plumbing diagram, you need to make the interior planning, design, structures, everything you have to design first. Now those designs are also further instruction manuals. Alright. There are engineers who will guide the construction of the building. There are supervisors who will manage the execution of the work. Laborers who will actually do the building. Enzymes are like laborers. Do the laborers become a part of the building? Now as the laborers are building this wall, some of the laborers became a part of the wall. Does that happen? No. So laborers are like enzymes. What will the laborer do? Whatever the engineer tells him to do. What will the engineer tell? Whatever the blueprint demands. Will the engineer tell the laborers to start creating modular kitchen when you have dug up the ground for foundation? When will the engineer tell the laborers to do the modular kitchen? When the civil work is over, no? Only when the civil work is complete, the engineer will tell the laborers to do modular kitchen. Where was the information until then? The blueprint was telling what needs to happen after what. Completion of something is the indication for the next thing to be initiated. You understand? So for our body also, blueprint is the DNA. Blueprint is the DNA. Enzymes are the laborers, workers. Hormones are like the engineers. Hormones are like the engineers who will activate certain type of laborers to carry out certain set of functions. There is enough material available for building anything. For building the entire building, there is a supply store. There is a supply store. When a particular laborer is activated, he knows what material to pick up to finish the job that he is supposed to finish. Now it is not one laborer, it is a set of laborers. It is a set of laborers activated by the engineer appropriate to the stage of completion of the building. Whatever is the stage of building is, the engineer will activate those set of laborers who have to carry out the next stage in the process. That's all. Have you seen Dalmatian puppies transform into adult Dalmatians? Do Dalmatian puppies come with black dots? Are they born with black dots? Have you seen even golden retrievers become adult dogs? You have seen a baby golden retriever and a puppy. Golden retriever puppy and an adult golden retriever. We have a golden retriever at home. He has become a bit old. Now 2 days ago while playing he has injured his hip dysplasia. He has developed some arthritis also. Anyways, it's like elderly dog now, but still child at heart. He wants to play. Anyways, the puppy will not be golden in color. The puppy will be creamish only, whitish creamish. If people who don't know that it will eventually turn golden will feel this is a duplicate dog. Not duplicate dog. It is not the breed that I want. Alright. So as the dog starts growing, at a certain stage, the golden color starts appearing in the hair. Part of the body is gold, part of it is white. Gradually it starts transforming, changing. Alright. Now even the growth of hair and nails and all that. It will grow to a certain extent. How much it should go, it should go. It will grow. It will not grow beyond that. It will not grow beyond that. The rate of growth of nails is not the same at all length of nail. When you cut it short, it grows faster. When you leave it, it will not grow as fast as it grew when you cut it. Now measure it. You can try. It might be a 2 millimeter nail that you cut. Calculate the number of days it takes for the nail to grow back to 2 millimeters. Let's say it will grow back to 2 millimeters in 7 days or 10 days. So if you don't cut your nails for 30 days, will it be 6 millimeters? It will not be. Then if you don't cut your nail for 2 months, will it become 12 millimeters? 1 centimeter. Like you are going around with, what is that? Vampire nails. Doesn't go like that. So there is always a signal, there is always a messaging. Hormones are like the engineers who will tell the laborers what to do. Enzymes are the laborers who will execute the work. You understand that? So metabolism is like the building process. Metabolism is the building process. Hormone is the regulatory process. It regulates who should work, how much should they work, when they should work, when they should stop, all that. So hormones will act on specific receptors. Enzymes will act on specific substrate. Enzymes will have substrate specificity. Hormones will have receptor specificity. Receptor is like the activation key. Alright. Hormone goes and binds to the receptor. There is activation of the mechanism of the production, whatever needs to happen. The set of enzymes which needs to be activated, when that key is pushed, will get activated and they will start working. Understand? So these laborers also start getting produced from the blueprint. When a hormone goes and activates a particular receptor, it is not that all the enzymes are already present in the cell waiting for the signal from the hormone. When the hormone gives the signal by activating the receptor, that will activate the blueprint to transform the blueprint into laborers. Laborers are being produced from the blueprint. You see that? That is when the gene translation happens. Transcription, translation, you see that, no? Transcription and translation happens when the hormone goes and stimulates the receptor. A particular receptor is stimulated, the transcription process is initiated in the DNA. mRNA is produced for the specific set of enzymes that needs to be activated for the process that the hormone initiated. Now this set of mRNAs will further go into translation process, protein synthesis. All the enzymes are proteins only, no? Except for a couple of exceptions. We can safely assume that all enzymes are proteins. Now these mRNAs are translated into proteins. Proteins further go into processing for final processing in where? Where does the final processing happen of these proteins? Ribosomes. In the ribosomes, the further processing happens. If necessary, it goes into the Golgi complex for further packaging. Gets used where it is supposed to be used. It's all a continuous chain of activity. When a particular hormone should be activated, that is also based on some signal. Imagine the body, whether it is human body or any other organism, as a very complex signaling system, a very complex system of feedbacks. Huge, massive. Just if you can visualize it, It is a universe in itself. So in our Indian philosophy, it is said that anybody who understands an atom understands the universe. Everything is interconnected. If you understand an atom in all its dimensions, you would have understood the entire universe. So that's why it is told macrocosm in microcosm. Cosmos is so macro, it is the same as what you find in the microparticle, that is the microcosm. An atom or a cell, every atom is a microcosmos. It is the universe in a miniature form. Alright. So when you study all this, a black hole if you study, stars become so dense and dense and dense and dense and dense and dense and nothing comes out of it. Very small size. And a nucleus is so small. When a nuclear fission happens, explosion that can destroy cities, 100-gram nuclear bomb might destroy the entire city. Where is so much energy coming from? Nuclear fusion, when two nuclei fuse, when nuclear fusion is happening, sun is burning so hot. Nuclear fusion is happening in the sun, no? So that is the vastness of coordinated action that is going on. Hormones are therefore significant. Plants also are working like that. Plants also have feedback mechanism. They also know when to produce what. So it's all a chemically coordinated set of thousands of chemicals interacting with each other. The intelligence in life is so humongous, not just limited to this. This is a part of the intelligence that is there throughout. Every cell is intelligent. It knows what to do, when to do, how much to do, what to respond to, what to not respond to, how to protect itself, how to attack, how to defend. Every cell is equipped with all that intelligence. Alright? So, see, from plant to Big Bang to sun, nuclear fusion, nuclear fission, universe, microcosmos, macrocosmos, everything. It's all connected. Everything is connected to each other. There is nothing that can be seen in isolation. Alright? Five hormones we see in plants. Now this one. So we will come back to plants and see through our ignorant eyes the limited functioning of what these chemicals can do. Alright. So how much ever we study, we will not be able to understand anything in its entirety. So we should always be humble. We should feel that, okay, I have tried to understand a little bit, I know a part of it. But no matter how much I learn, I will never be able to understand it completely. If I understand it completely, then I have nothing else to learn. No purpose to stay on this planet. My job is done. I will be gone. Alright. Ignorance is what keeps us alive. Okay. Complete realization of existence has no reason for existence in a limited form. As long as you are ignorant, you are alive. Alright. So nobody who is alive is completely enlightened. Anyways. So that is why it is told ignorance is bliss. You can stay happy, not knowing. Gibberellin is the hormone that will cause seed germination, initiate the germination of seeds by mobilizing the stored food. Gibberellins are plant hormones. I'll read through. Okay. There is not too much of explanation required for understanding the complexities. Coordination between different hormones is important. While studying plant hormones, we need to understand what the hormone is, where is it produced from, what does it do, what all does it coordinate with. They cannot work in isolation, no? In tissue culture we see auxin cytokinin difference. If there is more cytokinin, what will happen less compared to auxin? If there is more auxin compared to cytokinin, what happens? You want the plant to grow tall, what do you have to give? If you want more lateral branching, what needs to be given? All the hormones can be produced. So based on what the demand is, the plant will produce hormones in a balance. in different proportions. So we need to see if different hormones are in different proportions to each other, what will happen. So that will give us a fair idea of what hormones will do to the plants. Okay. Gibberellins are plant hormones that play a crucial role in seed germination. During germination, gibberellins stimulate the production of enzymes like amylase, which breaks down stored starches in the seed into sugars that the growing embryo can use as a source of energy. This mobilization of food reserves is essential for the seedling to grow. We discussed in one of the previous sessions, seeds are loaded with food. Plants load their seeds with food to ensure that the embryo gets some energy during growing stage. When the embryo is growing, it needs stored food to be utilized. You cannot have photosynthesis when the embryo is growing from the soil, from inside the soil. Only when it comes out and its green part starts coming up, then it can do photosynthesis. Until then it has to survive. Food should be available. But the problem is, this food should remain as unutilized food until it is stimulated. You don't want the utilization of the food to happen. So you have to keep the enzymes which can convert this stored food into energy inactive. But when the conditions become favorable, you have to make the enzyme get activated so that it will convert this food into energy that the embryo can use. But seeds are thrown away as soon as they are formed. Mother wants the seeds to grow in a particular way. Mother is telling the seed, see, mother is telling the embryo, you have to wait for the right conditions. When the conditions are right, you have to start converting the food that I have given you into energy, use that energy and grow. When you come out, you can do photosynthesis and you can still grow. If nobody eats you up, then you can still continue to grow. If you are allowed to grow to a particular size, then probably you will survive. All the best. Good for you if you survive. Good for the ones who eat you if you don't. Okay. So, this is what should happen. But it cannot be based on conscious awareness. It has to be based on chemical messaging. What do you do? If you are a plant, what do you want to do now? You have produced the embryo. Embryo is in a dormant, embryo is in a particular stage. You don't want the embryo to grow further. You have to tell it to stop. You have been produced to a particular extent. You have to stop. Wait for conditions to become favorable. The food and embryo to grow. The seed should start using the food and embryo should start growing. So two things should happen in the seed. That means two chemicals are loaded in the seed. One chemical which will not allow the seed to germinate. One chemical which will not allow the seed to have metabolic activity. It will put the seed in metabolic dormancy. When the conditions are favorable, this fellow who is making the seed dormant has to be removed, and the other fellow who will make the food available for the embryo should get activated. So two hormones are loaded into the seed. One hormone is seed dormancy hormone, which is abscisic acid. Abscisic acid is loaded in. So when abscisic acid is removed, gibberellin is activated. Gibberellin gets activated, which will stimulate the enzymes. I told you hormones are like switches, activation switch. It will activate the switch of production of amylase and other enzymes, which will break down the stored food and convert that into energy. Now the embryo is happy. It's getting food. It will start eating and it will start growing. This is how it goes on. So two hormones I told you. Two hormones associated with seed germination. One is abscisic acid, which will prevent seed germination. The other one is gibberellin, which will stimulate enzymatic breakdown of stored food, which will allow the embryo to utilize the food and grow. Alright. So, yeah. Amylase is an enzyme which will act on what? Amylase enzyme acts on starch. Amylopectins, starch. Again, I have given it in the form of table. Hormone, where is it produced from? Type of hormone, action on the root, action on stem, action on leaves, action on fruit, action on seed or germination. That's all, no? Major functions of plants. What will the hormone do to the shoot, to the root, to the leaves, to the fruits and seeds? This one table will give you complete details of what all you have to remember with hormones. Auxin, where is it produced from? Shoot apical meristems, young leaves. Auxins are produced from shoot apical meristems and young leaves. What is the type of hormone auxin is? Auxin is a growth promoter. What kind of growth it will promote? We will see. Action on root, promotes root initiation and growth. Auxin will cause initiation of roots and growth of the roots. On shoot, on stem, what does it do? Promotes cell elongation and inhibits lateral buds. Okay? So auxin is the one which will cause apical dominance. If there is more auxin, lateral growth will not happen. Lateral branching will not happen. It will make the plant grow tall. What will it do to the leaves? Promotes leaf growth, delays leaf abscission. What is abscission? Breakdown. Okay? Abscission is breakdown. What will promote leaf abscission? What will make the leaf fall off? See, the leaves are strongly bound to the stem. Correct? At its base, it is strongly held. They are all having cell wall. So the cell wall is strong and persistent. If you make the cell wall at the junction weak, then the junction will become softer and the weight of the leaf will be enough to for the leaf to detach from the plant. What are the layers of plant cell wall? Okay, let us not deviate. Let it be. Abscission is making the junction of making the junctional cell walls weak and making the leaf fall off. That's fruit also. Why does a ripe fruit fall from the tree? Why not a raw fruit fall? Some people will say because ripe fruit will be more heavy, sir. That is why it will fall. Logical, no? Unripe fruit is not so heavy. Ripe fruit is heavy, that's why it will fall off. No. The reason is the junctional, wherever it is held to the stem, the junctional cells will start, cell walls getting degraded. The cell walls of the junctional cells will start getting degraded, making the weight unbearable and it will fall. Alright. So auxin will cause leaf growth, delays leaf abscission. Fruit delays fruit ripening. Auxin delays fruit ripening. Who promotes fruit ripening? Ethylene promotes fruit ripening. Auxin delays fruit ripening. Then action on seed or germination promotes seedless fruit formation. Auxin is also involved in embryogenesis. For the formation of embryo, auxin helps. With auxin's influence, seedless fruits can be produced. Now you know, if you want to make some plant produce seedless fruits, you can experiment with auxin. Alright. Gibberellin. Where is it produced? Gibberellins are produced in seeds, young leaves and roots. Nature of hormone is excuse me. It is a growth promoter. Gibberellin will prevent root growth, inhibit root growth at high concentrations. Gibberellin will inhibit root growth at high concentrations. In that sense, gibberellin will act in contradiction to auxin. Promotes stem elongation. Gibberellin also promotes stem elongation. There is a disease because of excessive gibberellin production due to a fungus. The name gibberellin comes from that fungus. What is the disease? Bacani disease. Foolish seedling. Foolish seedling or foolish sapling disease. Continuous, rapid, long, tall growth and it does not produce anything. Grow fast, fall fast, die fast. Die fast, okay. So if somebody Somebody, some actor or some sportsman suddenly gets fame, they start behaving too arrogant, they start throwing around their weight and starts showing off that they are the biggest people, they will fall so rapidly only. Okay. There used to be a cricketer, I told you, I think I have told you. Have I told you? I don't remember what I tell where. There used to be two cricketers, school friends, who started their cricketing journey together. Both of them were part of the school cricket team. Both of them were top class batsmen, openers for their school team. They have a record of 600 run partnership for their partnership. Both of them entered international cricket, represented India almost at the same time. One of the two was a very aggressive batsman and shot to a lot of fame. Did not manage his anger, arrogance well. Started getting into fights with everybody. Was too arrogant and proud about his talent and ability to play. Did not believe that it is necessary to be humble. Did not realize that it is necessary to be humble. His career got cut short after 1996 World Cup. He did not play after 1996 World Cup. So both of them started almost in the 90s. Late 89-90 was when they started, both of them. The other one continued to play till 2011 World Cup. One is considered as a wasted opportunity, disgraced cricketer because of arrogance, and the other one is considered as the god of cricket. Who am I talking about? Sachin Tendulkar is one, god of cricket is easy. Who is the other one? His friend? The other one was a left-handed batsman. He was considered to be more talented than Tendulkar in terms of his stylish play, aggression, the kind of scores he made, multiple double centuries he had made then. Another left-handed West Indies cricketer became to be another legend. Brian Lara. Who is this left-handed cricketer who I'm speaking about? Sachin Tendulkar's close friend once upon a time, friend from school days, but unfortunately had to get out of international cricket because he was kicked out of international cricket. Got into addiction, got into bad company, a lot of things. Unfortunate. Who is he? No? You don't remember? Do you remember a cricketer by name Vinod Kambli? None of you remember. Even the next two generations will remember Tendulkar. My generation of people have seen Vinod Kambli play. So we remember. Wonderful player. There used to be talk about Vinod Kambli for many years after he was kicked out of international cricket. About 5-6 years ago, in an ordinary movie, he played the role of a sidekick of a villain. Survival necessity. And his story could have been such an inspirational story because he was orphaned at an early age. He was orphaned at an early age. He was taken care of by some charity institution. He grew well. They only funded his school education. He grew to prominence, but because he did not manage his hormones well, he had to fall. Foolish sapling disease. Gibberellin effect. Okay. The fungus of arrogance made him develop the hormone of gibberellin so much that he grew fast. Initially it was the aggression which led him to grow, but because of increased influence of gibberellin, aggression transformed into arrogance and he had to fall. Alright, gibberellin. Story. I like stories. Alright. Cytokinin produced by roots, transported to other organs. Cytokinin is produced by roots, transported to other hormones. It is the growth promoter, promotes lateral root formation, promotes cell division, delays senescence in shoot, promotes chlorophyll synthesis in leaves, delays leaf aging, delays fruit aging, promotes nutrient mobilization, promotes seed germination, works with auxin to regulate cell division. See, cytokinin and auxin interplay is what is utilized in tissue culture. Increase cytokinin, you will have more shoot production. Increase auxin, you will have more root differentiation, goes on like that. Cytokinin will cause lateral growth. Auxin will cause apical dominance. So increase cytokinin, it will counter apical dominance. It will not allow the apex to dominate. It will stimulate the lateral buds. So plant will decide when what kind of growth should happen, depending on the environmental conditions also. Abscisic acid produced in leaves, stems and roots, growth inhibitor, inhibits root growth at high concentrations, inhibits stem growth, promotes leaf abscission, promotes fruit drop, induces seed dormancy, inhibits germination, promotes stomatal closure in leaves, inhibits leaf expansion, induces seed dormancy, promotes desiccation and tolerance. Basically, every form of destruction is mediated by abscisic acid. Seeds will not grow, leaves will fall off, shoot will not grow, root will not grow, fruit will be suppressed. Why should we have it? Balance. You cannot have only one directional growth. You need to have checks and balances. Abscisic acid is also important because it creates a check and balance in the other growth promoters. You cannot have only growth promoters. You need to have growth inhibitors also. Only with growth promoters and inhibitors, you will have a balance of growth. Growth promoters are like accelerator in your car or in your bike or in your cycle. Growth inhibitors are like brake. Can you not have brake? If you look at only brake, if you look at brake as something that will stop your pace, then it will appear like brake is a problem. But if you look at the journey, it is not only speed that matters, it is ensuring that you complete the journey and reach the destination. Brakes are very important. Without brakes, there will be accidents. Same with life also, same with living processes also. You need to have something which will induce growth. Parallelly, you need to have something which can stop growth. If you can, our systems also, we have sympathetic system, we have parasympathetic system. Sympathetic system will stimulate, increase activity of various systems, various organs. Parasympathetic system will bring it down. Sympathetic stimulation will increase heartbeat. Parasympathetic system will decrease heartbeat. Imagine what would have happened if heartbeat once increased continues to beat at the same pace. Can you survive with heart being in your mouth all the time? No. It has to go back. It has to go back. Parasympathetic influence. Right? Sympathetic stimulation will help you to counter crisis. Increased activity of the vital organs will help you to counter crisis, help you survive in times of danger. Parasympathetic action will help you perform in a balanced way longer. If you want to travel longer distances, if you want to have a longer journey, you need to have a very well functioning parasympathetic system. Sympathetic stimulation is automatic. Whenever there is a crisis, sympathetic system is associated with survival instincts. Whenever there is a threat to survival, real or perceived, sympathetic stimulation will happen. If you feel something bad is going to happen, if you imagine something bad is going to happen, suddenly your breathing will become fast. Your heartbeat will become fast. You will start sweating. You will start feeling afraid. You will start feeling anxiety. That can happen. Can you do the reverse of it by thinking? Can you do the reverse of it? If this moment, if I take you through some small guided thought process, I can make all of you to start develop that anxiety. Alright? If I start talking about how your parents are struggling to ensure that you do well in your studies, the break that you have taken for a year, some of you more than 1 year, what if you are not able to clear this process and get into a medical college? The thought of being humiliated by people around, the thought of being feeling useless. See, everybody's faces are already changing. It's easy to get into that sympathetic stimulation mode because it is inherently designed, it is evolution designed to ensure survival. Parasympathetic needs practice. I cannot say, if I start telling you, imagine you all go through the process, and all 48 of you get 720 out of 720, and nobody can accuse you of rigging the paper or paying for getting the paper. That will not create the same kind of effect in your mind. Correct? It will not lead to a Zen-like feeling. Oh, I am floating. That doesn't happen. That requires practice. That is why the morning 45 minutes of practice that I have given you. Yoga and pranayama will give you control over your parasympathetic system. Sympathetic system responds automatically to real or perceived threat, to past stressors or possible stressors of the future. Parasympathetic system needs to be controlled. You have to take control of that. Pranayama will help you take charge of the parasympathetic system. So you can bring down anxiety when you want to. Breath is the only one which you can use to control the hyperactivity of the mind. Alright. So balance. Abscisic acid is important. Ethylene, produced in most tissues in response to stress, aging or ripening. Ethylene is also a growth inhibitor, inhibits root elongation, promotes root hair formation, promotes stem thickening, inhibits elongation, accelerates leaf senescence. Senescence means what? Aging. Senescence is aging. Promotes abscission. Abscission means breaking off. Senescence is to age, abscission is to break off. Abscisic acid gets its name from the hormone which will make the organs break off from the plant body. Promotes fruit ripening. Example, bananas and tomatoes. Breaks seed dormancy in some plants. Influences germination under stress conditions. Seed dormancy is what? Seed dormancy is the seed not being able to germinate. Seed being in a state of suppressed germination. It will not be able to germinate. That is dormancy. Dormant state is a resting state. See, you have seen, when you go out into fruit stalls. Okay. Ethylene will, as I already told you, hormones are like activation switch. They are like activating certain switches. Ethylene will stimulate the production of certain enzymes which will act on pectin in the cell wall, the cellulose of the fruits, different complex carbohydrates in the fruit, and slowly start breaking those complex carbohydrates and converting them into simple carbohydrates, simple sugars, fructose and all those sugars. That process will lead to sweetening of fruits. It will make the fruits sweeter because the pectin in the cellulose is broken down by pectinase, adding sweetness to the fruit. Complex carbohydrates are not tasty, they are not sweet, they are tasteless. So when these enzymes that are activated because of ethylene influence will start breaking down these complex carbohydrates. And ethylene is not suddenly produced in large quantities so that overnight the fruit ripens. Natural ripening of fruits is a slow process. It takes time. Gradually it starts ripening. You can see the color change slowly. You can see the aroma change slowly. As these complex carbohydrates and the complex structures are being broken down, it will lead to the production of aromatic chemicals also, which will give the aroma for the fruit. But have you seen fruit stalls where you have mangoes stacked up? All mangoes are appearing bright yellow. They are all having very nice color. Even if you are going and standing in front of it, you don't get the aroma of mango. Have you noticed it? They all appear very neat. It's like painting of mango. All the mangoes are of the same color. Impossible for a set of fruits to be at the same stage of ripening simultaneously. So these are all artificially ripened fruits. Artificially ripened fruits. What do they do? There are certain chemicals which will have similar influence as that of ethylene. Ethylene will cause ripening. Ethylene will also cause change in color. The chloroplast will get transformed into chromoplast. Chromoplast is colored pigments in the chloroplasts. You put a few drops of these chemicals in a bucket of water, dip these fruits and keep it aside. Dip and keep it aside. Dip and keep it aside. So the surface of the fruit has been exposed to chemicals which influence the fruit in the same way that ethylene influences. Bananas, you would have seen, banana ripening chambers will be there. You will get agarbatti like sticks, incense sticks, agarbatti like sticks which will release ethylene. Put all the bananas in the room, close the room, light this agarbatti which will release ethylene gas releasing agarbattis. So the room gets filled with ethylene gas. Bananas have peel, no? They have peel. Ethylene is acting on the surface of the banana. The surface of the banana, the peel of the banana is being acted upon. So the peel will start turning yellow. Ethylene did not penetrate into the fruit. Inside, the banana is ripening at its own pace. Inside, the banana is ripening at its own pace, but on the outside, you have stimulated the ripening process. So the color of the banana changes because ethylene is a gas and the cell membranes and cell walls are not airtight compartments. Some ethylene would have gone inside also. So some ripening would have accelerated inside also. So it is not completely unripe. But what is inside does not match what is outside. Outside it looks very yellow, bright yellow. It appears like the banana is fully ripe. Very tasty to eat. But when you eat it, you will feel that it is still raw. Alright. So what ethylene does is that it will stimulate the production of certain enzymes which will trigger breakdown of complex carbohydrates into simple sugars. That will cause ripening of the fruit. Right. Ethylene done. Okay. Summary. Auxin primarily involved in cell elongation, root initiation and delaying leaf abscission. Gibberellin key in stem elongation, seed germination and promoting fruit growth. All the things that we already discussed. So with gibberellin is directly involved in breaking seed dormancy and triggering the processes that allow the stored food in seeds to be mobilized, making it the correct answer to the initial question about seed germination. Whatever question we had about seed germination, this is gibberellin is the correct answer because it is what breaks seed dormancy. Coordinated functions, auxin and cytokinin. Now I want you to know what will happen to hormones in combination. What will happen to the plant when hormones work in combination. Alright. Auxin and cytokinin. Auxin promotes cell elongation, particularly in stems, and is crucial for root initiation. Cytokinin promotes cell division, particularly in shoots and leaves, and delays leaf senescence. Alright. In coordination, what do? The balance between auxin and cytokinin determines the overall pattern of plant growth. High auxin to cytokinin ratio promote root development. More auxin, root differentiation. More cytokinin, shoot differentiation. So in tissue culture, when the callus is formed, you will start varying the auxin cytokinin ratio. You will first increase the auxin levels, root differentiation happens, then decrease the auxin level and increase the cytokinin level, shoot differentiation happens. And then you separate that from the callus, put it for hardening, then the plants are ready for distribution. Very important questions come on this. See, in plant hormones, auxin, apical dominance, gibberellin, elongation, plant elongation, ethylene, ripening, fruit ripening, gibberellin breaks seed dormancy, important. Abscisic acid promotes seed dormancy, growth inhibitor, very important growth inhibitor. Cytokinin in relation with auxin, differential growth, suppresses seed apical dominance. So opposite of auxin, both auxin and cytokinin are growth promoters only. They are both growth promoters. But cytokinin has an inhibitory effect on auxin. They are different kind of growth promoters. Auxin will promote apical growth, cytokinin will promote lateral growth. So if you allow auxin, it will not allow lateral growth. If you allow cytokinin, it will not allow apical growth. They are like two different political parties. Both will say they want to do development only, but the idea of development for both political parties are different. Alright, one political party has a particular idea of development, the other one has another. Both are good for the country, no problem. But if these fellows come, they will not allow this kind of development to happen. If these fellows come, they will not allow this kind of development to happen. We should be happy that whoever comes, some development is happening. Alright, so plant is happy that whether it is auxin or cytokinin, some growth is happening. Alright. So growth cannot be one dimensional or one directional. It is always a balance of multiple factors. Alright. So plant is a great example of democracy. Everybody has a say. Depending on who is powerful at what point of time, their programs will get prominence. But fortunately for plants, it is not once in 5 years things will change. It keeps changing dynamically based on the surroundings. Alright. Apical dominance is auxin. Gibberellin and abscisic acid, seed germination and dormancy. We already discussed. Gibberellin will promote seed germination, it will break dormancy. Abscisic acid will induce dormancy. They are opposite to each other in seed germination. Gibberellin breaks seed dormancy and promotes germination by stimulating the synthesis of enzymes that mobilize stored food reserves. Abscisic acid induces and maintains the seed dormancy, particularly under unfavorable conditions, and inhibits germination. Balance between gibberellin and abscisic acid regulates the timing of seed germination. Very important. If the seed germinates when the conditions are unfavorable, the likelihood of the embryo to survive is lesser. So seed should germinate when the conditions are favorable. So the chances of survival for the embryo will be higher. You cannot assure survival. When you soak seeds to make sprouts, you have your stores, retail stores, let us say. If the business is going on well, it makes sense to expand the stores. But suppose the economy is going into recession, there is a bout of pandemic, COVID is happening. It doesn't make sense for you to increase the number of stores. Rather, you might have to decrease the number of stores. You may have to shut down a few stores which is likely to not get you any profits, or which is likely to push you into more expenses. So you have to shut down. When the conditions become favorable again, you again start. Alright. So business, when done as per natural principles, you do business with as per natural principles, you will have good business. Nature is a good business coach. Nature is a good life coach. Nature is a good relationship coach. Nature is the coach for everything. So if your principles of doing anything is based on the principles of nature, it will be very, very effective, because nature has developed these principles by evolution. We will be developing it only by our imagination or our own understanding. But if we align it to nature, then it will be more productive for us. Ethylene and auxin. We already discussed. Ethylene, fruit ripening and leaf abscission, promotes ripening of climacteric fruits. What are climacteric fruits? Like tomatoes, bananas. Seasonal fruits or sweet fruits. Shedding of leaves. Auxin delays abscission by maintaining cell wall integrity in the abscission zone. As fruit matures or leaves age, auxin levels decrease in these tissues, making them more sensitive to ethylene. Auxin and ethylene are acting in counter to each other. Auxin is holding on to the cell wall integrity. Auxin's influence will make the cell wall remain intact. As the leaves age, auxin production comes down. As the fruits become more and more aged, auxin production comes down. Now these same tissues which were under the influence of auxin, maintaining its intactness of cell wall, becomes more responsive for ethylene. Alright. It's like somebody has two friends. One friend is having a strong influence of this fellow for a long time. But over time, this friend makes a certain distance from that friend. Another friend gets an opportunity now to influence. Ethylene will do that, causing abscission. Ethylene promotes the thickening of stems as a response to stress, while auxin involved in the directional growth, gravitropism and phototropism. Gravitropism is also referred to as geotropism. Trophic movements are what? What are trophic movements? Trophic movements are directional movements. Movements that happen in response to certain stimulus. What stimulus? Light, water. Geotropism and hydrotropism. Geotropism, hydrotropism. Geotropism is movement towards the soil. Okay. Ethylene, cytokinin, gibberellin in combination. Ethylene promotes leaf senescence and abscission. Cytokinin delays senescence. Gibberellin promotes overall growth and delays senescence. What will happen when they are working in coordination? The coordinated action of these hormones determines the timing and progression of senescence. When should the leaf age? For example, during fruit ripening, ethylene accelerates senescence, while cytokinins in young leaves or fruits can delay it. See, hormones can travel through transportation mechanisms, correct? You want only the fruits ready for ripening to become loosened, to become weak. Senescence should come, then abscission should be initiated. But the fruits which are not yet ripe, which are not yet fully grown, should not get influenced by ethylene. Correct, no? But you are not releasing ethylene only there. There is a possibility of ethylene coming to the surroundings also. So as long as this auxin, cytokinin or gibberellin is influencing the leaf or the fruit, ethylene will not be able to influence it. The influence of these growth promoters will come down as age progresses. So when the influence of the growth promoters or protectors of senescence or those who prevent senescence come down, the one who can initiate and induce senescence and abscission can influence. You get it? In movies you see, no? Somebody is being protected by security. As long as the person is being protected by security, there is no possibility of attack for somebody who wants to attack. The person who wants to attack will look for what? The timing when the security is weak. Same. Consider ethylene to be the assassin trying to assassinate a gangster, and these growth promoters are like the security guard. Security guard is protecting them, and when this security cordon becomes weaker, the assassin gets an opportunity to hit. All right. Cytokinin and abscisic acid goes on like that. Cytokinin and abscisic acid interact to fine-tune the plant's response to stress during water scarcity. Abscisic acid induced stomatal closure happens. Cytokinin will maintain nutrient mobilization. See, the range of questions that can come from growth hormones or plant hormones, not too wide. Their functions, coordinated action. Remember abscisic acid wherever it acts. Stomatal closure, abscisic acid helps. So remember stress. When there is stress, abscisic acid will help you overcome stress or manage stress. No water or lack of water supply is a stressful situation. So if you want to ensure that you conserve water, what is it that you want to do in a plant? Prevent the loss of water. How do plants lose water predominantly? Through transpiration. if you want to prevent transpiration during water stress, what do you want to do? Close stomata. So if the question is, which is the hormone that can bring about closure of stomata during stressful situation? It is abscisic acid. It is abscisic acid. Alright. So those kind of small principles you remember, you will be able to answer questions. Done. These things enough. We will read out the conclusions. The interplay between plant hormones is complex and finely tuned to ensure growth, development and responses to environmental stimulus occur in a coordinated manner. Alright. The balance between growth promoters like auxin, gibberellin and cytokinin and growth inhibitors like abscisic acid and ethylene ensures that plants grow optimally under favorable conditions while conserving resources and surviving under stress. These hormones in hormonal interactions are context dependent, meaning the effect of one hormone can be influenced by the presence and concentration of another as well as by the specific environmental conditions the plant is experiencing. When you see all this, doesn't it become very obvious that the plant is also trying to survive and reproduce? Correct? When will you survive better? You survive better when you are able to manage the influences from your surroundings. There are influences from the surroundings. Every organism is being influenced from all around. The organism has to respond to the influences, adapt to the influences in such a way that the organism develops mechanisms to survive in the best possible extent in the given circumstances. That is dynamic survival mechanism, no? Survival is not static. It is not like if this is happening, this is the response. No, it is dynamic. Dynamic survival mechanism is, you know that you have to survive. Every cell of your body knows that it has to survive. To ensure that, do whatever is appropriate in a particular context. And it happens automatically. It happens automatically. Body is designed to adapt that way. Body is designed by millions of years of evolution to adapt and acclimatize that way. Alright, that is why when you are exposed to, let's say, heat, body sweats, and the sweat spreads on the entire skin. It forms a coating on the entire skin, and that coating will act as a water film on the body, using the heat from the body to get evaporated. As the heat from the body is being taken away, after sweating you start feeling cooler. There is a mechanism to overcome every situation. Overcoming the situation is not possible. Adapt to the situation. If there is a possibility, get away from the situation which is threatening your survival. That is what we do. It's raining heavily. Now if you have to move from point A to point B, you will try to find out ways by which you don't get drenched. If there are no such ways, you will try to cross in such a way that you are drenched to the minimum. If there is sunlight, you try to take shade. So the responses are designed automatic. Plants also are doing the same thing. Bacteria also are doing the same thing. So in any given condition, under any given circumstances, whatever is the best thing to be done will be done in a balanced way so that the response is appropriate to ensure better survival. That's all. Everything will come down to that. Improve the possibilities of survival, increase the chances of reproduction. How does that correlate to humans? How does that correlate to humans? In a non-human situation for any other organism, survival is dependent on what? Think about other organisms other than human beings. Then we will see what happens to human beings in terms of survival. Then you will know all of that you are doing and everybody is doing is for survival only, in different ways. How do we ensure survival? How does an organism ensure survival? Or what leads to better survivability for an organism? Better survivability increases the chance of reproduction. Every organism wants to find a mate who has better adaptability to survive, with an evolutionary hope that their offspring will also survive better. It's all subconscious. That is why, okay. Anyways, we'll come there. What increases the chances of survivability? What increases the chance of survival for any organism? We're talking about survival. What increases the chances? Staying awake increases your chance of survival in my class or in any class. All right. Okay. Now, what increases the chance of survival? Unfortunately, this room has a podium and I am tall enough to see. Okay. Don't sleep. There is a subject in first year MBBS called pharmacology. Pharmacology is completely, you will be dealing with chemicals, drugs, interactions and all that. Very dry subject. And every day during our first year MBBS, afternoon 2 to 4 is pharmacology class. Post lunch, 2 hours. And the class will always be taken. There were many people in the department of pharmacology, but our HOD always wants to take the class. He takes the class every day. 2 hours. One good thing about him was he will not make the people who sleep in his class uncomfortable. He doesn't mind. Alright? And he used to be so monotonous in his speech. He's very entertaining also. He used to make a lot of attempt to keep the class entertained, but pharmacology was very boring for me also. Second year MBBS, not first year MBBS, second year MBBS. Alright. Some of our classmates got used to sleeping in that time, 2 to 4. It became a habit for some of them. Like some of them would start sleeping even before our HOD comes. And they wake up when attendance is being taken in the end. They wake up, they present sir, and then they continue to sleep. So I don't know whether they have been able to get out of that habit yet, because it was a very powerful habit that our pharmacology HOD had put in them. Alright. Unfortunately, you don't have those luxuries. You will be made to feel very uncomfortable. Alright. Okay. What increases the chances of survival? Not in the class, general, for organisms. Ability of food. Okay. Expand the scope a little bit. Food is fine. Food is correct. But expand the scope a little bit. Don't say food. Give me something else which will include food also. Resources. Access to resources. You have better access to resources, your chances of survival is high. Yes or no? So why do organisms build their nests and have their habitation around sources of water? Access to resource. Correct? The first important adaptation that one needs to do is have better access to resources, increases your chances of survival. Now, the rest all there are evolutionary mechanisms, adaptations, physical adaptations, physiological adaptations and all that. When it comes to conscious adaptation, evolutionary adaptations are there. But consciously what every organism tries to do is, every organism will try to get a maximum share of the resources. Or a share of the resources ensure comfortable survival for them. Desires and reality will not always match. Everybody wants that. Everybody wants access to resources. But the resources are not enough to cater to everybody's wants. Resources are enough for everybody's needs, but everybody operates from the standpoint of want. Even in organisms, in animals also, in creatures also, wherever there are resources, there are more organisms trying to get access to the resources. When there is attempts to get access to the same resource which will help them improve their chances of survival, what will it lead to? Competition. Competition will lead to conflict. Conflict will lead to complex social interactions. And complex social interactions will make life a struggle. Alright, every organism. It all stems from the desire to have access to resources, which is coming from the evolutionary drive to ensure survival. Because better survivability is linked to better reproductibility, the ability to reproduce. For most organisms, the possibility of reproduction is dependent on the availability of a mate. Correct? Unless there is a mate available, the organism cannot reproduce. Selection of mate is dependent on resource access. A particular organism which cannot prove to its mate, and in nature mostly the one who selects a mate is a female. Female selects the mate because female is the one who has the power to reproduce. The female is designed evolutionarily to identify a mate who can be a contributor of genetic material leading to the production of strong offspring. That is why you see in nature male animals fighting to impress a female animal. The female animal is watching. Come on, what can you do? Rejected, get lost. Alright? It is evolution. It is evolutionary. It is designed that way. Nature has designed organisms that way. It is not conscious. It is by default. It is by default. The more naturally aligned an organism is, selection of mate is more appropriate. Alright? So, extrapolate this to the human situation. What are you trying to do? Why do you want to become doctors? Want to save the world? You need access to resources. Human survival is not just dependent on access to food and shelter and water. Human survival is dependent on access to different levels of resources. Physiological survival dependent resources, access to food of your choice, access to shelter of your comfort, access to facilities that you want to feel good about yourself. Human beings being social organisms, access to those kind of resources which will help you build proper habitation in where your near and dear ones are safe and happy. Evolutionary drive. And your conditioning, your experiences and your observations have made you feel that becoming a doctor will give you access to resources which can make your life better and which can make you capable of surviving better. Rest all the things are there. Human beings can think beyond survival. Human beings can think beyond survival. So all the other things are also there. But the inherent drive should match with that. And some people will threaten their survival and do things which are generally not related to survival. They are also seeking some other kind of resources. The resource of social acceptance, recognition, reward. Those are all resources in human societies. Human societies have complex multi-level resources. Access to different levels of resources will give you different kinds of power over your surroundings. Based on the kind of power that you have over your surroundings, your ability to survive and your ability to influence your surroundings change. You understand? So everything comes down there. At the baseline, we are no different from any of the other organisms that we find on the planet. But we are not like any of the other organisms because we have awareness of what's going on. Every other organism lives according to what's going on. Evolutionary drives drive the organism's existence, but human beings have an awareness of what's going on, have an intelligence to understand why things are happening the way it is happening, and have the power to decide, plan, determine and work towards a particular outcome of their choice. No other organism does it. A tiger hunts when it is hungry. When the tiger is not hungry, even if the prey is next to it, it will not do anything. It will just sleep and watch the small baby deer play. It will say, nice, nice, nice. Wait here till I get hungry. All right. A snake bites. A snake bites when it feels threatened. When it feels that its survival is in threat, biting is an escape mechanism for the snake. It's a defense mechanism for the snake. No organism will prophylactically kill. Oh, there is something going around here. It might harm me someday. I'll kill it. No other organism does it. No, it's only human beings who do it. Right? No other organism kills that it nurtures. I'll tell you the extent of cruelty of human beings. No other organism kills that which it nurtures. Meaning, if an animal takes care of some other animal, nurtures it, provides it love and care, gains its trust, it will not kill that to eat. Human beings do that. Yes or no? What is sheep rearing, poultry farming, cattle farming? The animals are nurtured to kill. Animals don't know that. Correct? The animal doesn't know that. A sheep, when it is a small baby, somebody play, it's nice to play with the sheep. When you are playing with the sheep looks up to you like a baby looks up to the mother. The emotions are pure with animals. When the baby is playing with somebody, the baby connects to that individual or organism as what it is supposed to connect to. It doesn't know. Even if suppose I have nurtured a sheep and it has grown from a baby to an adult sheep. Now the sheep has to be killed. If I carry the sheep to the slaughterhouse, the sheep will not even doubt it. It might even think that it is a tour. Till the last moment, it will not even realize that I have nurtured it to kill, and it will die in shock. Why? What happened? Probably he did not notice that I was being killed. Okay. Humans have misused their intelligence. Humans are not natural in their emotions. Okay. Humans are worse than carnivore animals. Carnivore animals are predictable. Correct, no? You know what the tiger does is known. What a snake does is known. Only unpredictable, dangerous animal is human. Alright? Anyways. So plants also are like that. Alright? This is the question. Which of the metabolites is common to respiration mediated breakdown of fats, carbohydrates and proteins? We already discussed this while discussing glycolysis, Krebs cycle and electron transport chain. Acetyl-CoA, glucose 6-phosphate, fructose 1,6-bisphosphate, pyruvic acid, acetyl-CoA, all of these are participating in multiple pathways. Acetyl-CoA is important because it can be a byproduct of the breakdown of carbohydrates, fats or proteins, and it can be a product for the synthesis of carbohydrates, fats and proteins. All of these are interchangeable. Breakdown of glucose is called what? Glycolysis. Breakdown of lipids is called lipolysis. Breakdown of proteins is called You have some metabolic pathways for energy production. Glycogenesis, glycolysis, gluconeogenesis, lipolysis, lipogenesis. If you are breaking down proteins, you can call it proteolysis. But proteins are being broken down for the synthesis of energy. You call it gluconeogenesis. That is, glucose being formed from the breakdown of proteins or lipids. Lipolysis is breakdown of lipids. Lipogenesis is production of lipids. Acetyl-CoA is something which can go in any of these. Glycolysis leads to acetyl-CoA. It can lead to acetyl-CoA by converting pyruvic acid into acetyl-CoA. Acetyl-CoA can be used to produce glucose also. Reverse process is also possible. You break down lipids, you get acetyl-CoA. Use acetyl-CoA, you can make lipids. Alright. So it's like when you have too much of glucose or carbohydrates, acetyl-CoA gets produced. You don't have regulation of how much of glucose is broken down into pyruvate, and all of that is happening. Cell will not utilize all the glucose that is available. Whatever is in excess will get converted into lipids. Acetyl-CoA, if it is not going further into the Krebs cycle for further ATP production, there is saturation, gets utilized, pushed into storage. So acetyl-CoA is an intermediate which goes into any of the pathways. Any of the three major metabolites have carbon, hydrogen, oxygen. All lipids have carbon, hydrogen, oxygen. All proteins have carbon, hydrogen, oxygen, nitrogen. In addition to that, there can be more. There can be others. Alright? Like phosphoglyceride, fructose phosphate. Is it a carbohydrate? Is fructose phosphate a carbohydrate? In glycolysis we see glucose 6-phosphate. Is it a carbohydrate? It is. The nature of a chemical, what it is dependent on its primary structure. Phosphate is a secondary functional group attached to the primary structure of glucose. So it is still a carbohydrate. When we look at glucose 6-phosphate as a carbohydrate, now this particular carbohydrate has phosphorus also attached to it, has phosphorus in it. So what else can be there along with the primary elements? That is different. But primary elements are common to all. Carbon, hydrogen, oxygen are common to all the macronutrients. Nitrogen is available in various forms because of it is stored in the muscles also, various ways nitrogen is available. So when you have the elements available, what you need to build needs an enzymatic machinery which is already there in the body. The enzymes will restructure and build it. Acetyl-CoA is one of the intermediate compounds which can be used for building a lot of these macronutrients. You understand? Just important metabolites and which pathways it can enter and what is the purpose of the pathway that it is entering into. Some simple tabulation I have given. Metabolites of glycolysis and Krebs cycle that enter the other pathways. Glucose 6-phosphate, where is it found? Glycolysis, we know that it is there. It can enter the pentose phosphate pathway. It is also called pentose phosphate shunt. Production of NADPH and ribose 5-phosphate for nucleotide synthesis and reductive biosynthesis. See, our same glucose 6-phosphate which is there in glycolysis can get into a pentose phosphate pathway, ribose production, nucleotide synthesis. Ribose is also a sugar, no? It is also a carbohydrate. So pentose phosphate pathway is useful for producing nucleotides. Glucose 6-phosphate can get into that also. Pyruvate, seen in glycolysis, pyruvate can go for amino acid synthesis, serves as a precursor for the synthesis of certain amino acids like alanine. Excuse me. Citrate, different intermediates can go into different pathways. I have given the list of those which are significant, which can come as questions. Citrate is found in Krebs cycle. Citrate can go for fatty acid synthesis. You see, our glucose metabolism intermediaries, one is going for amino acid synthesis, one has gone for nucleic acid synthesis, one is here, this fellow is going for fatty acid synthesis. Everything covered. Protein, pyruvate goes for protein synthesis, citrate goes for fat synthesis, glucose 6-phosphate goes for nucleotide synthesis. Alright. Citrate is exported to the cytosol. Cytosol is cytoplasm, where it is cleaved to acetyl-CoA for fatty acid synthesis. Citrate is formed how? So how is citrate formed in Krebs cycle? Acetyl-CoA plus oxaloacetic acid. The reverse is also possible, but in Krebs cycle we have mentioned that reaction to be irreversible. Here citrate being converted to acetyl-CoA means that the reaction of Krebs cycle is reversed. Nothing is absolute as such. If it is continuing in the Krebs cycle it will not be reversed. But if citrate is available, citrate can be taken away. Citrate can be taken away. Krebs cycle is happening where? Krebs cycle is happening in mitochondria. Acetyl-CoA comes, oxaloacetate comes, citrate is produced. Let's say 10 molecules of citrate is produced. 2 molecules are taken out into the cytoplasm. Who will stop? You can't stop it, no? 2 molecules get into the cytoplasm. This enzyme, citrate synthase, will not reverse this. Aconitase will take it off. Citrate will be taken by aconitase to convert it into alpha ketoglutarate. Citrate can be taken out into the cytoplasm. If citrate is taken out into the cytoplasm, some enzymes related to fatty acid synthesis can break down citrate and release acetyl-CoA, which can be pushed into fat synthesis. Alright. Alpha ketoglutarate, Krebs cycle, amino acid synthesis. Precursor for the synthesis of glutamate, an important amino acid. Oxaloacetate, Krebs cycle, useful for gluconeogenesis. What is gluconeogenesis? Production of glucose by using substrates which is not carbohydrate in nature. Used in the synthesis of glucose from non-carbohydrate sources during gluconeogenesis. Okay. What do you call the mechanism of production of glucose from carbohydrate sources? So let's say if you break down glycogen to make glucose, what do you call it? Glycogenolysis. Lysis of glycogen, glycogenolysis. But if you break down fats and convert it into glucose, what do you call it? Gluconeogenesis. When the beginning of the process of glucose production is not from a carbohydrate source, you call it gluconeogenesis. Gluco is glucose, neo is new, genesis is formation. Formation of glucose from a new source, which is a non-carbohydrate source. That is gluconeogenesis. The difference between gluconeogenesis and glycogenolysis should be very clear. You should not confuse between those. Alright. Next is succinyl-CoA. It's a product of Krebs cycle. Heme synthesis. Succinyl-CoA is a precursor in the biosynthesis of heme, a component of hemoglobin and cytochromes. You see this? Heme is a component of hemoglobin, that we know. Heme is also, cytochromes are also iron containing compounds. Cytochromes are also iron containing compounds. Heme is present in that also. Precursor is coming from Krebs cycle, succinyl-CoA. So blood formation from the byproduct of glucose metabolism, important. All right, clear? Everything can be referred to somewhere else. Every metabolite in the glucose metabolism pathways, glycolysis and Krebs cycle, goes into something else. Something else from somewhere else they can come and join this process. Okay, it is not like citrate which has been produced by citrate synthase only has to come into this. Another citrate can come and join. Citrate synthase cannot say, hey, I did not produce, you get out, you cannot go further. No. All that the enzyme requires is a substrate. Where the substrate is coming, it doesn't matter. Alright. Alpha isocitrate dehydrogenase will convert isocitrate into alpha ketoglutarate. Where this isocitrate came from is of no concern to isocitrate dehydrogenase. Understand? If glucose 6-phosphate is available, fructokinase will convert it into, fructose 6-phosphate is available, phosphofructokinase will convert it into 1,6-bisphosphate, fructose 1,6-bisphosphate. It will not ask whether you came from glucose. Did you come in this line? No, doesn't matter. So enzyme requires substrate, it will convert it into product. Enzymes are like cold killers. There is a saying in Kannada, "Aaru kotre atte kade, moor kotre sose kade." Alright. The eternal conflict that you see in family melodramas, serials, is between who and who. In melodrama serials that you find on television, the serials will become popular if the conflict between A and B is the crux of the serial. Who is A and B? Huh? Correct, tell. Mother-in-law and daughter-in-law. Mother-in-law is called atte in Kannada. Daughter-in-law is called sose. So the saying goes like this. The saying is, somebody in between, let's say this is mother-in-law, this is daughter-in-law. This is the enzyme, mediator. Alright? If this is more, if the mother-in-law gives me more money, I will talk for her. If daughter-in-law gives me more money, I will talk for her. Enzyme is like that. If there is more substrate, if there is more of A, it will convert A into B. If there is more of B, it will convert B to A. That's all. It's like a mediator. It can do both ways. Alright? If you are unmarried, it can get you married. If you are married, it can get you divorced. If two people are separate, enzyme can bring them together. If they are together, it can separate them. Ultimately, enzyme needs to have job. Like lawyer also, no? Marriage lawyer and divorce lawyer, same. So when he conducts the marriage, he will congratulate them and tell them that if you need any further help, you can come back to me. I can help you with divorce also. Enzyme is like that. Alright. Okay. Other metabolites from other pathways entering glycolysis or Krebs cycle. Glycerol, see, lipid metabolism, fatty acid and glycerol is produced when lipids are broken down. Glycerol from lipid metabolism, purpose of the originating pathway, why was it produced? For breaking down fats to provide energy. Where will it enter? Glycolysis. It can be converted into glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate can continue further to become pyruvate. So glycerol can enter the glycolysis. Fatty acids, they are in the lipid metabolism, they provide acetyl-CoA for production of energy. They can enter Krebs cycle. Fatty acids in the lipid metabolism can be converted into acetyl-CoA. We saw acetyl-CoA getting converted into fatty acids. Reverse also can happen. and that can enter Krebs cycle to be further broken down and released as carbon dioxide. Lactate produced during Cori cycle, that is anaerobic glycolysis. It is for the purpose of regenerating NAD plus during anaerobic conditions. It can enter glycolysis. It can be converted back into pyruvate. Pyruvate can get into aerobic respiration pathway again, or pyruvate can get into other pathways. What did pyruvate get into when I told you earlier? Pyruvate can get into? Huh? What did pyruvate get into? Look at this table again. Where did pyruvate go? Just remember this. Glucose 6-phosphate is going in for which pathway? What is it going in for? It's going in for nucleic acid synthesis. Pyruvate is going for amino acid synthesis. Citrate is going for fatty acid synthesis. 6 we noted here: alpha ketoglutarate, amino acids, oxaloacetate, gluconeogenesis, succinyl-CoA, heme. Here some of them: glycerol for glycolysis, glyceraldehyde 3-phosphate. Then we have fatty acids, becomes acetyl-CoA, gets into Krebs cycle. Lactate produced from Cori cycle or lactate metabolism, converted back into pyruvate, gets into Krebs cycle. Amino acids, what produced amino acid there? Alpha ketoglutarate became amino acid. Amino acids can be broken down to give you alpha ketoglutarate, which can get into Krebs cycle. How many carbon is alpha ketoglutarate? 5 carbon. Fructose metabolism in the liver, alternative source of glycolytic intermediaries. It can enter glycolysis as fructose 6-phosphate. Alanine amino acid we saw. Alright. Amino acid metabolism, don't worry about the Cahill cycle. Transports ammonia from muscle to liver. It can enter glycolysis and get converted to pyruvate. Enough. Alright. So, metabolic pathways are interconnected. In all this, you can remember those small things. What can get converted into what can get into what. This is the information which can help you answer questions which most of the students will not answer. Not so difficult to remember also, because it's a small summary tabulated content that I am given. So you can remember it. Advantage information that gives you advantage in the exams. So to cross beyond 650. Alright. So yeah. In this discussion on the metabolites, intermediaries, you should understand that nothing is absolute in the chemical equilibrium, in the chemical environment of our body. Nothing is absolute. Everything is interlinked. Anything can become anything through many things. That's all. Anything can become anything through many things. That means you can convert anything into anything, provided you know what are the things that will be in between. And is it the only pathway to reach there? No. Many pathways. It's like again a spider's web. Like the knowledge that you have to build. One point A to point B will have multiple pathways to reach it. Chemical balance in the body is also like that. The knowledge you build is also like that. It has to be like that. If it is like that, then you will be able to do well. Alright. Okay. So, we still have about 2 more questions. One question. Which of the following statements is correct? How many questions we are left with? Now this is again plant reproduction. Okay. I'll tell you these 4 terms. Testa, tegmen, tectum, tapetum. What are these? 4 terms. Sounds nice to hear, lot of rhyming, all related to plants, but confusing. Testa, tegmentum, tapetum. Alright. So these kind of things are also there. So similarly appearing things which will confuse you. Okay. Plants of the developing pollen grains obtain nourishment from tapetum. That was the question, no? So tapetum. Seed coats are referred to by the terms. The outer hard seed coat, the inner not so hard seed coat. Testa, tegmen. What is tectum? Everything related to that only. Testa, tegmen, tectum. Testa and tegmen are a part of seed. Tectum is the outer layer, sculpted part of the pollen grain. Tapetum is in the pollen sac, the innermost layer of the pollen sac. So if you see testa, tegmen are female structures. Tectum, tapetum are male structures. All four are related to plants. All four are related to flowers basically. So some connection to the flowers. Anyways, we'll come to that. So the question is related to tapetum, no? The question that next question we'll be dealing with is tapetum. Where do the pollen grains get their nourishment from? Then do we have another question? Perisperm and endosperm difference. Okay. We'll come. 2 more questions. Out of the 9 questions, we have dealt with 7 questions successfully in just 3 days. Okay. 3 days and just 9 hours. But we have been able to deal with topics which will take weeks to deal with. All right. So, anyways, okay. Congratulations for not listening to your bladder. You can treat your bladder with a break now.